Okay, good morning, everyone, and welcome back to the 5th Annual Plant Science Symposium. For those of you that were unable to join us yesterday, my name is Julian Janori, and I'm the current president of the Plant Science Council here at the University of Florida. Yesterday, some among you participated in the Breeding Games workshop to try to create the best yielding cultivar for the production of craft beer. Other fellow students participated in the annual poster competition, which had to be adapted to fit this online format. Award winners for both of those competitions will be announced later today at 2:50 p.m. Eastern Time, right before our closing remarks. We have a fantastic lineup of invited speakers today that I'm eager to learn from, as well as student speakers who were chosen to present their research at this symposium. Before all of that, it's important to note that none of this would have been possible were it not for Corteva Agri-Science. As mentioned yesterday, these symposia provide opportunities for the next generation of scientists to explore their interest in the plant sciences by inviting and hosting public and private experts on the topics of our choosing. There have been one man in particular that has been alongside of us every single step of the way throughout this planning process, and he's here today to kick off day 2 of our Plant Science Symposium. So I would now like to actually introduce Dr. Jason Rauscher. Go ahead.
Great, thanks, Julian. Can you hear me okay?
Yep.
Excellent. All right, I'm going to share my screen here. There we go. Share and there. Okay. So it's a pleasure to be here today in a virtual format, joining you at the University of Florida for your symposium this year. I was trying to think back through the last year, and it's a little foggy. It might be that way for many of you, but Florida was one of the last in-person events that we had last year in the Symposia series. So I'll talk a little bit more later about the impact of the pandemic on the Symposia series, but it's a pleasure to be participating today in the symposium, and looking forward to a great day. So Rauscher, I'm Academic Relations Manager in the R&D. And the Plant Sciences Symposia series is an important program within our, sponsored by our company. This is I just got a unstable internet connection, so let me know if I freeze. So this is a Corteva-sponsored global program of scientific symposia initiated and organized by graduate students. So as you'll see here, we have the Plant Science Council, who have been doing a great job organizing and putting this together. And we believe this is really a high impact model for supporting graduate education around the world. And it really truly is around the world. We are on 6 continents, at 16 countries and over 60 institutions since our founding back in 2008. And so we have a great international, usually in an in-person event, that's a huge component of it. On these virtual events, we do try to recreate it. And I know the students had a networking session yesterday, and I think they've really done a great job trying to continue this networking even in this virtual environment. So I really want to thank the students for all their hard work there. Career development, it's very important to us to have both public and private sector participating. A Corteva speaker. Our speaker today, Sarah Lira, will be participating later in the day. Whatever term you want to use, and being ready for whatever career they choose to go for after graduate school. So speaking of leadership and teamwork, I took this from their website. The Plant Science Council is really a spectacular group with great leadership, and I've been very impressed by this group and their ability to put together this both this this symposium and all the other great work that they do. So we're really very proud of these students, and we think that the Plant Science Council is really organization. Which has brought together multiple universities across Brazil that are associated with the Plant Sciences Symposia series. And so they've been doing some great work. I'll talk about that in a moment. And then we also have our Student Advisory Council. This is a council of students representing different institutions right now in the US and Canada are represented. And as hopefully you can notice there, we do have a representative from the University of Florida. Christina is on our council, and this is a great group. We had a meeting yesterday. We have some great things lined up, hopefully to really leverage this international network and help institutions that participate in our program really get the most out of it. So look forward, especially this year, to really seeing some great things out of this group. Now, one of the things that they did last year, well, actually, I'll get to that in a moment. So this is typically what one of these symposia should look like. Lots of people gathered together. This is an image that looks very foreign to us at this point in time with the pandemic. We know that it's had a profound impact on academia and industry alike. And we just want to give a great thank you to all the professors and students who have adapted to the situation and all the online courses. And it's been a big year. We really are coming up on a year here of the impact. So we really appreciate all the work that has been put into this. Now, as I mentioned, that Student Advisory Council, one of the things they did was they responded to the kind of the early stages of this pandemic by putting together a symposium themselves. So these were representatives of multiple institutions on this council, and they put together a plant breeding symposium back in April of last year that was very successful. It had over 1,000 participants from countries all over the world. It was quite international. So we were very proud. If you want to learn more about this, there's a publication on it in CSA News in July of last year. And then Intergen, as I mentioned, they did something similar. So they put together a Brazil-focused symposium that had over 900 participants from an incredible number of institutions around Brazil, over 55 universities and 80 other public and private institutions in Brazil. So it was really an important event, a two-day event back in July. And so we're hoping that this year as well, we'll continue with this tradition, and even moving forward, once we start having in-person events again, having some of these virtual events that really have that global reach, I think will be an important addition to the Symposia series. So last year we had lots of, well, not a lot of cancellations, but a lot of some postponements. Most of the obviously since about March, all of the symposia were converted to virtual format. And we continue that way. We still don't have an idea of when we will get back to in-person events, but here you can see the virtual events that we have coming up in the next couple of months. After Florida, we have Texas A&M coming up on the 18th. You have two choices on February 18th. There's also the Phenotyping Conference, annual conference, 2021. And they have a early career symposium there. University of Missouri coming up, University of Hawaii. Unfortunately, we don't get to travel to Hawaii, but they are having a virtual symposium if you want to feel like you're there. And then Ohio State, and we're going to have in Brazil, the Association of Seed Technology there is part of their annual conference, which has been postponed to later in the year. But throughout this year, they will be holding a kind of a seedathon, kind of a hackathon solving problems of the seed industry in Brazil. So if you're interested in that kind of thing, please, and if you speak Portuguese, that'll be a great event. And their kickoff is going to be on March 29th. So for more information, hopefully soon we will have these updates on our webpage at symposiaseries.corteva.com. We do have them updated on our Plante network. Please join our Plante network if you haven't. You can follow this URL here and become a member of the Plante and get updates, and hopefully we'll be using that quite a bit this year. And also, if you use LinkedIn, don't hesitate to use the hashtag Plant Science Symposia, so we can see your comments and your participation in these symposia. So again, thank you. We appreciate everybody who's participating today, and we really appreciate all the students and the faculty advisors who have helped to make this happen today. So I will hand it back to Julian.
Okay, thank you, Jason. And of course, thank you, Corteva, for allowing this event to happen, virtual or not. So our first presenter of the day is someone who I've heard a lot about from my first day as a PhD student here at the University of Florida. I heard how much Dr. David Clark has achieved as a plant breeder and a researcher, so when I set up a meeting with him as a first-year student, I was understandably nervous. I remember walking to his office and seeing all the awards on the wall. His group was the first to produce genetically engineered plants with ethylene-insensitive flowers that last twice as long as normal flowers. Building on success, his lab engineered plants to not only produce larger flowers, but also to produce leaves that remain green longer, and to enhance floral fragrance. His work in functional genomics has led to the development of the largest publicly available DNA sequence database in the world for his model species, Petunia. Dr. Clark has since expanded his research group to include an applied focus on tropical plants, developing a conventional breeding program for the production of new varieties. In the past decade, his Coleus breeding program has now released several new varieties of colorful annual breeding plants that are now growing in summer gardens across the world. I remember sitting down and the first thing he asked me was, "What is the ploidy level of your plant?" Dr. Clark, I don't know if you remember that, but I certainly do. I froze because, to be honest, I wasn't 100% sure at the time. I thought to myself, this is going to go terribly. As our conversation progressed, it became easier and easier to talk, and I left that meeting feeling motivated to succeed in my program. The reason I bring up this little story today is to highlight that, as successful as he is, Dr. David Clark has the patience to guide, mentor, and inspire students. And it is with that in mind that I'm happy to introduce Dr. David Clark.
Wow, thank you, Julian. That was a very nice introduction, and I do remember asking you that because I think that's probably the most important first question to ask any grad student about their crop is what's the ploidy. So anyway, I'm going to go ahead and share this screen and get started because I've got a fair amount of ground to cover this morning. Let's see. Okay, is everybody good? Julian, you see that?
Yeah, you're good to go.
Okay, thank you. Okay, so first off, today I just wanted to thank the Plant Science Council. It's an amazing group of students, and I've seen the whole thing develop. And in particular, I wanted to thank Julian and for his fine leadership, and Sammy for organizing and getting me involved in this. Corteva, thank you. Esteban and Marcio, just everybody, thank you very much for being here. It's good to see the participation. I'm not going to be talking about big data sets. I know that that's the focus of what this conference is about, but when I was asked to talk, I was asked to talk some about my Coleus program, and I realized that I really wanted to focus on the students as my audience and hopefully put the philosophy into PhD and show you how to make a program successful from the bottom up. And this is all stuff that I've gained from experience. So I'm going to be talking more about the left side over here, the common sense side today with my talk. A little bit about me. I usually don't talk about myself too much, but it's relevant to today's lecture. This is a picture of me when I was an undergraduate. I grew up in East Tennessee, and I trained in horticulture for all three of my degrees. I learned nursery production at the University of Tennessee when I graduated in '88. Went into post-harvest physiology and floriculture greenhouse production with a master's at Clemson in '90. And then late in my graduate life, I learned how to clone genes and do molecular biology, particularly focused on ethylene biosynthesis and getting into ethylene receptors. I didn't do a postdoc. I came straight to University of Florida, and luckily I had the tools. They wanted people that could grow greenhouse crops and also engineer plants to do new things. So that was that new burgeoning field of biotechnology in the mid '90s. So early on in my career, I did basic science. I worked with Petunia, inbred lines of Petunia. Worked on molecular biology and plant transformation because that's what I was mostly skilled at after I finished my PhD. I collaborated, the first collaboration I made was with Dr. Harry Klee. He's become one of my best friends and one of the best scientists I've ever known. We worked together on ethylene receptors and signal transduction to engineer long-lasting flowers and fruits. And on the bottom left, we have engineered petunias and normal petunias treated with ethylene. And then the same gene, mutant ethylene receptor from Arabidopsis, engineered into tomatoes at 0, 11, and 100 days after pick. And so this was what we were working on early on. I was building a lab. Harry exposed me to collaborators at Monsanto, and I learned a whole lot about business and how to go to market and all kinds of new things through that. And I was also learning how to manage people. In graduate school, we teach you how to clone genes, but we don't teach you how to manage people. So that was one of the hardest things that I had to learn early on. As I progressed through the middle part of my career here at Florida, kept with my basic model of petunia, but the ethylene work ran its course. We made a large cDNA sequence database and started doing microarrays to look for ethylene regulated genes. And we found a whole series of genes that were downregulated by ethylene. And we found out very quickly that petunia, when it's pollinated, it makes ethylene, and that triggers to the petals to stop making fragrance to attract pollinators. So that led to, I had very little experience in biochemistry, so I had to learn that. And we had a very good transformation knockout system. And we started knocking out genes in the volatile fragrance biosynthetic pathways. And so all of the different enzymes that catalyze these reactions, we were cloning all of those genes and then knocking them out in transgenic plants. At that point in time, I also started a little side project in coleus breeding, and I'll get to that a little bit later. But that's when you graduate up to, you learn how to manage people. Now it's more about mentoring and putting thought into the people, because the people product is really important to what we do. Here lately, and I don't say late or retire, but I am on the last quarter of my career. It's hard to believe that, but for the last 10 years, I've been applying the knowledge that I've gained over time to crop improvement. So I've moved more from a basic focus to an applied focus, and you'll see over the course of this talk why that's happened. Now my focus is to create and maintain a global coleus cultivar release pipeline, and I'll talk a lot about that today. We're breeding new crops. I'll talk a little bit about basil, and I won't talk much about hibiscus. So we keep adding new crops to our program. The focus now for me is on philanthropy, giving my time and my resources and spending time with people like Julian to sit down and just talk to them and see if there's any way that I can help them change their lives in a positive way. So my word cloud has gone more toward helping with my time and my resources to try to help other people, because after this long in the business, it's not about me anymore. It's about the next generation. So one of the things that I do, and I stick to this very strongly, is I live by the S curve. This is a typical S curve. We've all seen this. We start out in making a new program or anything that we're going to do, and it takes us time to get into the learning curve. We get into an exponential part of the curve where everything is clicking, and then the science or the program will mature over time, or you get bored and want to go into something else, and it starts to flatten out. And so this early part of the curve is really hard work, and getting the value out of the last part of the curve up here is also a lot of hard work. So what I try to do is stay in this sweet spot right here and keep it as long-term as possible, because that's where all the gain is. Now, for people that are young, like the graduate students that are going to be going out in the industry or going out into academia, the standard curve is the black one in the middle. But if you work harder, or you get lucky, or you're really smart or whatever, you can shift that curve to where you can extract value out of your projects quicker. If you delay it, or if you're, unfortunate or whatever, it will go to the right. And that's, on the left, when you make the curve shorter, you get promoted, you get tenure, you get all the things that come with it. If you don't do that and you go to the right, then you go to the NFL, which means not for long when you work like that. So that's going to happen whether you're in academia or in industry. Over time, you'll have this kind of thing happen with your career. And what happens is as you succeed and go to something else, then the expectations go up and you try to keep going out of the box and keep going up. So this is what you hope to be the natural projection. And my molecular biology went to biochemistry of fragrance, and now lately I'm in breeding, and I may have one more S-curve in my career. So the title of the talk was Good, Fast, and Cheap, How to Make a Program Successful. And you may have heard of this concept before, but I'll just review it for you. In most things in life, there are good, fast, and cheap, and you only get 2 of 3. If it's good and cheap, it won't be fast. If it's good and fast, it won't be cheap. And if it's fast and cheap, it's usually not good. And it's thought that this middle box right here is impossible, or something like a unicorn that doesn't exist, or utopia. So that's something that's very true about a lot of things. Now, to make a successful program and try to approach the 3 of getting good, fast, and cheap, you have to manage your assets well. So I'm going to talk a little bit about managing assets in business here for a sec. So the main assets, and this is in retrospect, that I have to manage are time, which is your most valuable asset, infrastructure, which is the most limited and neglected asset. Labor is obviously now, to me, the most difficult asset because everybody that you manage is different. And technology is the most obvious asset. You guys are all high-tech people, and you are driven by the technology, or you wouldn't be doing what you're doing. But the truth is that all of these change every day. Okay, the technology changes, the labor changes. Okay, and all of it is fluid. Now, in managing time, this is your most abused asset, especially as you age and you get old like me. I'm 55. Your time gets more expensive and more limited. It's a supply and demand thing, right? We always think about airtime, but you also have their time, which is your staff and the people, your collaborators. And there's a tendency to underestimate the value of their time. Okay, so it's really important to take time to train people, and that's not very productive time. And you also have to manage opportunity cost on their time, meaning if you have them doing one thing, what are you missing that they're not doing? So you have to pay attention to that. And especially in academia, you have to assume that your labor force is always transient. Okay, so even your staff will likely go on, if you train them well, to better jobs. If you miss certain time, if you miss time limits, you miss opportunity, and time is never cheap. Okay, so that leaves good and fast. Now, with managing infrastructure, it's always limited and it's always neglected. If you're in academia or if you're in industry, the infrastructure that you use is not always yours. Okay, so you have to share it with other people, and that causes a lot of confusion. It's often, because it's shared with others and not always yours, it's often neglected because it's not necessarily your responsibility or any one single person's responsibility. So the tendency is to run the infrastructure until it runs out of gas. Infrastructure, never cheap. So that leaves good and fast. You have to manage labor, and to me, labor is the most difficult thing that I manage because I was never trained to manage people. And I came out of college and came to University of Florida and had to do that very intensely. I believe in strength finders and Briggs-Myers test to understand what my staff is capable of. Me, I'm ENTJ. I'm a commander, bold, imaginative, strong-willed. My PhD student, Sammy Burrell, is ESTJ, organized, punctual leader. And there's times where we just don't understand each other at all. And so when you know your staff, you can play on your strengths and cover your weaknesses by covering the board. And to me, that's something that I'm always trying to achieve. You've got to lead by example, get in there and get your hands dirty. You got to learn from your mistakes. And when you fall off that horse, you've got to get back on it and ride very quickly. And so labor, never cheap. So that's leaving good and fast. Now, technology, all of you are technology nuts, right? So that makes technology the most obvious asset. And technology follows that S curve that I talked about earlier. It's good, you learn it, and then you get exponential with it. And then all of a sudden you may be paying somebody else to do it because it's cheaper. That technology is what attracts us to science. But over time, I've just learned that technology is just a tool in your toolbox. Okay, I started out as a molecular biologist, and that's how I introduced myself. But over time, I've had to be good at genetics, biochemistry, physiology, informatics, horticulture, stats, pathology, entomology. And that really leads to why you've got that PhD, that it's not doctor of horticulture, it's doctor of philosophy. So you've got to think your way through all this. You're going to be paid to think for a living. And if you can't do all of these things that you need to do yourself, then you have to collaborate to cover your weaknesses or hire that. And so it's not cheap. And again, that leaves good and fast. So you also have to manage business with a program. You have to know your stakeholders and your partners because their problems influence your program. Okay, you're responding to something that somebody wants out there when you're breeding a new plant. They want more yield, they want a better color or a better fragrance or anything. You are the solution. Otherwise, the companies wouldn't be coming to you, or the funding agencies. And their success is a reflection of your success. So a lot of times the companies or the organizations may get more billing of the success than you will. But the bottom line on managing businesses is that you've got to manage the supply chain and understand the supply chain of anything that you're involved in. Because if you ignore that supply chain, you're going to ignore opportunities. And this supply chain is what drives the selection criteria in your breeding program. You may be breeding for longer lasting tomatoes, and you're at a different point in the supply chain than getting better yield. And so you need to know the whole thing. Because if you're getting better, longer lasting tomatoes, they still have to produce out on the farm at a level that a normal tomato would produce. You also, lastly, have to manage value, and you have to know your currency. And over time, I've gone from doing all basic science to now more applied science. And the currency changes. As a basic scientist, you want to crank out good manuscripts, and you want to get grants, and you want to have your people be successful, maybe go to graduate school or go on to better jobs. In applied science, it's more about cultivars and intellectual properties, patents. You may be looking for research contracts with private companies instead of an NSF or a USDA grant. And all of those are valuable currencies. And again, even in the applied world, we have to make good people, because without them, we don't get anything done. Now, the one thing that I have learned, and I have had a big basic science program and a big applied science program at the same time, and it's really hard to do both of them well. Okay, so you cannot dabble. You have to understand that dabbling is going to waste time and space and all of your assets, whereas exploring has a whole different attitude, and that's when you're going out there with a real purpose versus just going along and trying to do 2 or 3 things at the same time. Okay, so realize that you have to really focus and sometimes you have to make choices whether you're going to do one or the other. Now, ultimately, you have to be a wizard, because you're going to go from good, fast, cheap, and impossible in the middle on the left to good, fast, and cheap and making it happen. You've got to try to fill the black box, and you're not going to do that by being this type of wizard like the Wizard of Oz. It's more like plant wizards, okay? And one of the things that I've realized, and this may be just a reflection of me being an older man, but I go back now and I look at people like Luther Burbank. He was known in his day as a plant wizard back in the late 1800s to early 1900s. Lou Gim Gong is the Florida citrus wizard. And I go back and I look at how these people who their philosophies have lasted throughout time. I realize how little technology, how little infrastructure, how little people support that they had in their time, and they were still able to make all of this happen. Okay, so I encourage you to go back. Sometimes I understand you guys stay in the current literature, but go back and read about these people. And I think that you might understand a little bit more about what I'm talking about being a plant wizard. See, plant wizards, plant breeders are an eccentric group. They're usually not mainstream. They love plants. They have vision because they realize that genetics is what allows them to predict what's going to happen in the next generation. They're observers and they have good eyes. They look for little things. They keep secrets. They don't tell everybody everything that they know because they realize that that's competition. They're very frugal and efficient, and usually miserly, and they work hard. And usually they merge art and science together. So this drawing on the right is my interpretation of what the normal plant breeder that I know looks like. And so they're usually a very eccentric lot. Now, so that said, that's enough of that philosophy. I want to talk to you about this Coleus breeding program because that's what I was invited to come and talk about today. And hopefully you can see during this process how that philosophy relates to this program. So this is my office. And this project started with one graduate student, Penny Wynn, back in 2003. So it's 18 years ago. And so Penny wanted to breed plants and not clone genes and make transgenics. She was on a fellowship because she was extremely smart. And so she didn't want to clone genes, and that's where we were with my program at that time. I asked my mentors from Penn State and Clemson what they thought about that, and they encouraged me to go that direction because they pointed out that many conventional plant breeders were retiring and programs were being turned into molecular biologists. And they said, you know, down in the future, we're going to train a lot of molecular biologists and then we're going to need breeders again. So we did what any good starting program would do, and we got 4 potential crops that we were interested in. And we started gathering as much genetic variability as we could to screen and see if it was going to work for us. We wanted to see if there was a lot of genetic variability. What was the seed to seed time? If it was going to take 2 or 3 years, then we couldn't produce a PhD project. And we wanted plants that were easy to grow because we didn't need cultural complications. So those were the main things that we were looking for in a program. So there were ultimately 3 losers and 1 winner. We looked at Scutellaria, skullcap, Plectranthus, which is very similar to Coleus, and Zephyranthes, which is a bulb known as rain lily. We threw out Scutellaria because there wasn't enough genetic variability. We threw out Zephyranthes because it was hard to grow. You had to basically flood it to make it flower, and we didn't think that that was good, and it was a bulb, so that was difficult. We didn't choose Plectranthus because we made some initial crosses, and everything that we evaluated out of that was truly ugly. So we gave up those and very easily discovered Coleus because of this amazing amount of genetic variability. It goes seed to seed very quickly, and it was something that we could grow very easily. So Coleus is actually a very recent plant. It's less than 200 years in domestication. It's from Indonesia and the islands surrounding there, India, in China, southern China, Malaysia. It was brought to Western Europe by the Dutch in the 1830s, and that's when they were down in Indonesia colonizing that for part of the spice trade. And so they would go around the islands and pick up interesting plants, go into Java to their nursery, multiply them up and send them back up to Western Europe. And that's exactly what happened. Here's a picture of Van Gogh's interpretation, Coleus plant in a flower pot from 1886. So it was already in people's gardens at that point. Here's a 1868 example of the variation. I still see this type of stuff today. So it was quite variable from the beginning. The name has changed a lot. It's now Plectranthus scutellarioides. It's in the Lamiaceae or the mint family, and you can usually tell those by having square stems. The positives about this plant, the most important thing is that there's amazing genetic variation. Julian, it's a tetraploid, okay, so that helps us a lot. But also, it's got at least one transposable element or jumping gene. We inadvertently sequenced a piece of one several years ago, and I suspect that there may be another one. The one that we sequenced is an ACDS type, and I believe that there's another one that's a retrotransposon type that jumps around. So every generation, we create new genetic variability. We go to seed one time per year. If we went quicker, and we could go 2 generations, but it's so much work, and we are selecting for late flowering, so we go one time per year seed to seed. It's very easy to grow and propagate by cutting, or by but not so much by seed, because we can't get inbred lines, and it scales up to fit the supply chain. So it was everything that we wanted it to be. There are some challenges, so not everything is easy in the coleus world. It's very hard to cross physically, and we get very low success with hand pollination. So the flowers are very small. If you go and emasculate and hand pollinate, you get about 10% seed set, and there's only 4 seeds per little flower, so it's really tedious. We've done it, but we use open pollination now. There is inbreeding depression. So you go about 2 to 3 generations, and all of a sudden, if you probably 98% of the stuff, all the families start to die out. Okay, so they get very, very weak very quick. If you're producing seeds, the seeds only last 2 years. You can't put them in the refrigerator, or you'll kill them overnight. So there's no cold storage. You have to keep them out at room temperature. Mother Nature can end this because we collect all of our seeds out of the field through open pollination. And so if we have a bad year, these plants usually start flowering right in the middle of hurricane season. And so we've had years where we didn't get very much seed production at all. And so if you have to rely on the previous year's seeds, you're really walking a tightrope. The other big challenge for us is growing our program in mixed growing facilities. We are in the greenhouses where teaching and research are mixed. And so sometimes people bring in plants with bugs and diseases. And we are particularly challenged by a virus called Impatiens necrotic spot virus, which basically means that we have to throw out the whole crop. And we had to do that in 2017, 2018, when that was brought in on some cuttings that went to a horticulture club and then spread by thrips. So it's very difficult to operate when you can't control. Remember, I told you about infrastructure. You can't always control what's going on. So as I said, you have to manage assets well. And so with coleus, if you look at time, it's a pipeline that runs 365 days a year. I mean, we never stop. We have slow times about 1 month a year. In terms of infrastructure, you've got to have a lot of space. And we have that. We have that luxury here at the PSREU down at Citra, and we've got a decent amount of greenhouse space, so that's not bad. Labor doesn't have to be highly trained, so we can use undergrad students and a staff member or two. And the technology that we use is absolutely not complicated. At the end of this, you're going to say, I can do this, and any of you could do this if you had these facilities. We use recurrent mass selection, which is the simplest breeding possible, where you maintain a population, you pick out the best plants and you collect seeds off of them, you grow them the next year, and you continue to do the same thing. So you just always pick the best stuff, and the population gets better over time. So by comparison to a lot of programs, coleus is absolutely fast and absolutely cheap. And the good part of it is my job. Okay, so I have to be the eccentric that's going to be able to have the vision to choose the good plants. You also have to know your partners. I'm very fortunate to have 3 partners: Ball Horticultural Company, Dooman Orange, and Proven Winners. These are their logos on the right. Ball gives us big box stores in North America, European Union, and Asia. Duman gives us big box stores in North America and the European Union, big box meaning like Lowe's and Home Depot. Proven Winners gives us high-end garden centers and more online sales in North America and the EU and soon to be in Asia. They all compete in the same markets. They all pay the same royalties, and they all get to pick from the same germplasm. Okay, so everybody knows they are direct competitors, and they all behave similarly, and we maintain our program transparently. And so we don't go to market with a new variety unless they ask us to. We never put anything out for bid. So these partners are really important because University of Florida does not grow plants and market and advertise and do all of these things. So you can't do anything if you don't have them. Now, our main research objectives with this program are very simple. First, we want to make tough, what we call idiot-proof plants. And what we mean is, if you recall that figure from the supply chain, we want to make it easy for producers. We want to make it easy for growers. We want to make it easy for the shipping and handling folks and the people at retail and marketing. Okay, we want all the way out to the garden. Ultimately, we want the gardener to get a good plant, and then it performs well in their environment, because we know that they'll buy another one next year. We want consistent bright colors and patterns, superior branching and architecture. You got to have good shape of plants. Late flowering is the most important trait, and I'll show you why here in just a little bit. And ultimately, our market is clonally propagated cultivars, not by seed. This is all by cutting. And most importantly is the people product. And we run a lot of students through this program to get them exposed to large populations and how to throw away plants, which is very difficult for young breeders to do. As I said, we use recurrent mass selection, which is the lowest tech of my career. Okay, I've been trained in a lot of things, but this is from the technology in very easily. As I said, we goes 365 days a year, and we collect open pollinated seeds. I'll show you where we do that. And we do trial seed planting in fall to look and see what particular seed families we want to collect. We only know the maternal parent because it's all from open pollinated stuff. This year we collected 650 seed families, and we ultimately planted 213 families with about 60 to 70,000 seedlings. So those are all up now. If anybody ever wants to come and take a look at them in the greenhouse, we're just starting this planting cycle. We will grow these plants for about 6 weeks until they look like the flat on the top. They look like a carpet going across. And at that point, I will go in and spend a week selecting down to 1,000 plants. So at that point, I will throw away 59 to 69,000 plants and only keep the best 1,000. And so that's where I make my money as an artist, because what I'm really good at is picking the right colors and shapes and architecture of plants early on. We do lots of selection. So typical type of selection is a flat on the left turns into a flat on the right. And we do over and over and over. I spend lots of time. You have to look at the plants at different times of day, because with different light, they have different colors. Ultimately, we throw away, like I said, probably 98% of the plants. They go to the dumpster. Oftentimes we will spray them with Roundup because we don't want anybody dumpster diving and getting our intellectual properties. So this is a very vicious selection right here. Ultimately, we will pot those up after 6 weeks and grow them until April. And this is what 1,000 plants looks like in my greenhouse. And we call this showtime. This is when the 3 companies that we work with come into our facility, and they make their picks of the plants that they want to trial. This year, we took pictures and did this virtually, and it actually worked out better. Each company saved anywhere between $5,000 and $10,000 in travel. They had more of their staff around the world picking their plants, and ultimately they got very good selections, and it gave us a photographic record of every plant. this is what it looks like when they come. This is from year before last. Each company has a different color flag: yellow, blue, red. There is jockeying for position, because if you come first, you get the first flags. If you come last, you get to see what everybody else is picking. Sometimes there are multiple flags in a particular container. And what we do is once we have all their picks, and they'll pick anywhere between 50 and 80 varieties to trial, we will sign material transfer agreements and then shear the plants and FedEx the cuttings to them overnight at their different rooting stations and trialing locations. We'll also then take cuttings for ourselves for our own trials in-house. And so we will take we'll select one more time and then go to the field. So our 2020 crop, this is last year, we took 750 what we call elite varieties or elite codes out to the field. And you can see, I mean, if it anywhere along the way, if it flowers, if it looks ugly to me, if I just don't like it, I just throw it away. So I have a wealth of genetic variability. Now, our field trials are down at Citra. We have sun trials and shade trials, both located down there. We take 750 codes and we clone several of each. This is our sun trial. We put 2 to 4 plants per code in the sun and then duplicate it. So we'll have 2 field plantings of this. And then due to limited infrastructure, we have 1 plant per code in the shade. In all of these trials, we take plant vigor, consistency, branching, flowering time data, and that's all on a 1 to 5 scale. We just give them 1 for bad and 5 for best, and anything in the middle, we stick it into an Excel sheet. We sum all the numbers up over all the trials over all the year, and then we just essentially sort it by the sum, and we throw away the lower 75% and keep the last 25%. So usually a couple of hundred different codes that we'll keep over. This is what the shade house looks like. It's about 0.2 acres, which is limited. These are 750 mature plants. This is where we collect our open pollinated seeds. We have honeybees on both sides, and they come in here and mix all of these different genotypes together. This is also more protected than out in the bare sun field, so we get better seed production, and it's about 30% shade. In the summer, in the sun field, this is what it looks like in the late summer, and it's a pretty ugly trial at this point, but this is where you learn the most. If you look at this trial in June or July, everything looks beautiful. But if you're looking in, say, October, it starts looking like this. And so it's very easy to pick out the good ones. Here's an example of what I meant by late flowering being our most important trait. Coleus plants, if they go to flower early, they mobilize their stored reserves from the lower leaves and send it to make the seeds. And ultimately, by the end of the growing season, they can rat out, like the ones here's 3 plants on the top right, 3 plants on the bottom left that are siblings to the plants right next to them. So it's pretty easy to walk through that field and pick out the really good ones. So again, there's no real technology here. We compare, we take pictures in sun and shade, especially things that look really good in the sun. And we go over and just walk over and compare them to the sun and shade. Here are 2 pretty plants, but it's the same plant. And if it looks like this in the sun and looks like this in the shade, we discard it, because when you're going to Lowe's or Home Depot, which photo do you put on the tag? If you put this shade figure and someone takes it and grows it in the sun, they'll bring it back or want a refund. So we can't tolerate that. Something like this, where it looks identical in sun and shade, will ultimately likely go to commercialization if it's got late flowering. And this particular plant was named Riverwalk, which is one of our more successful ones. I said that you have to know your partners. ultimately, it does split out to where they do become more important. Ball makes 54% of our business. Dooman is 36%, and Proven Winners is 10%. They go to market with named cultivars that have registered trademarks and receive U.S. plant patents, and that's how they market them. We sometimes name the plants, and sometimes they name the plants. It just depends on what their purpose is. What happens with a program like this is that there are some very valuable intangibles. If you get in Southern Living or Better Homes and Gardens with your varieties in the springtime, those particular varieties sell quite well in the upcoming year. So the companies will promote varieties that are the best ones with these garden magazines, and sales of those varieties go up. The coolest part of my job is that I get to go around the country, and everywhere I go, I can see the plants. Here they are in Detroit. Here they are in the top right in Portland, down at Epcot, and Churchill Downs on Derby Day in Kentucky for the Kentucky Derby. I can go to my hometown of Kingsport, Tennessee. That's my mom. She goes and takes me and shows me the coleus. I can go on Archer Road and see them down at Lowe's. I can travel in the summer. This is Chicago on the Magnificent Mile, one of the most important retail streets in America, and see them everywhere. Okay, and so what's really cool is I get to go and see my plants, and all of these people have no idea why I'm taking my picture in front of a coleus plant. But you know, it's kind of I'm a bit of a voyeur, and I love watching them enjoy my plants. Here is a plant, their best-selling plant, Redhead, which is around this white plant in the middle. And it was named after my wife down here, who is a redhead. And so, you know, it's kind of nice to be able to go through and see those sorts of things, and nobody knows that you did it. I get to travel quite a bit when we're not in a pandemic. I was called to go to London. One of our plants named Campfire made it to the final list of the top 20 plants at the Chelsea Flower Show. So I go over there. This is the plant, and what happens is it gets exposed to about 500,000 people in that week of the Chelsea show. There's Waldo here on the left wearing my IFAS colors, but there's Waldo in the back, right there in the back, walking behind Kate and Will. So it's pretty cool to be able to do that sort of stuff. What that leads to, again, Campfire makes it to the Chelsea list. Campfire makes it to Southern Living magazine as the best plants for that particular year, that in the spring. So it was number 2, and what that resulted is a doubling of sales of this plant in American markets. And so it's pretty, you know, I mean, you wouldn't think about this sort of thing being important in your academic or in your academic career, but it's very important in what I do. Last year, we had the distinction with Main Street, Beale Street, as producing the first coleus to ever be selected as All-American Selection winners, and that's the All-American Selections are an 88-year-old program in America, so we're pretty proud of that. Now, over 18 years, we're now on 4 continents. We've been planted in over 1 million gardens in America for each of the last 3 years. Our varieties have won awards in over 500 different trial awards, and there's all kinds of patents and cultivars released. So my currency has changed, but this has been a very productive program. The best bottom line on coleus is that our students have been very successful. Anybody that's been involved in it intimately has done quite well. It's good financially. We see that in 2020 we went over 100 million cuttings sold with our 3 companies. But the reality on coleus is that here's where we are, right? And you're just starting to see that it's getting into the part of the S curve that I'm not really comfortable with. So what's the next S? Well, if you have extra money from coleus, you can start exploring other crops. I work with sweet basil, Osmium basilicum. A few years ago, there was a basil downy mildew that was introduced into the Miami airport that spread across America. And we very quickly found out that Osmium basilicum has no resistance to basil. That's why it's hard to get organic basil now. We decided that we wanted to breed downy mildew resistance. So it starts with a couple of students, Adam Mosley and Shannon Bly. We go out and we find good culinary basils, tetraploids, and we find from the USDA grain accession a source of resistance with a wild Ocimum species that's a hexaploid. And we make the F1 hybrids. All were resistant, okay, so the resistance was dominant, but all of our hybrids were mules. They were self-sterile. 4x by 6x theoretically gives 5x, okay, and that doesn't work. We then take those plants and just put them out into OP like coleus with a bunch of other different basil selections. And we started a similar to our coleus breeding program, an open pollinated and then crossing regime to make clonal cultivars, vegetatively propagated varieties, because we wanted to be to market early. And so we knew that with our partners, we could make vegetative varieties, be first to market. And then during that time, we found out that we could backcross these F1 hybrids in one direction to the tetraploid. They ultimately ended up losing a chromosome and acting like tetraploids. And we backcrossed to get flavor, good flavor. And then we started selfing to break fertility barriers and make inbred lines for seeds, because that's how basil is normally propagated. So what happened? Well, this one plant, 1623-2, turned into a plant that our partner Proven Winners took to market called Amazel basil. I love the name. Amazel is on the left, and here is normal culinary basil on the right. These weren't inoculated. These were just grown at the time of year when the disease is particularly bad. So it's as close to completely resistant as possible. It's made it to market. In fact, it's so special that it got its own pot with Proven Winners, which is a big deal in my world. So it was released in 2018. It was for sale in 2019. And this is what it looks like down in Naples, Florida, with my coleus back there in the back paying for it. Martha Stewart sold out in 2019, and I heard that she sold out again in 2020, but I haven't been able to find that page. So ultimately, after Amazel, we started making the seed propagated inbred lines. That was started by Chloe Collins. She was an undergraduate. She's now a Huck Fellow at Penn State. And Sammy Burrell, your colleague, works on cloning resistance genes to try to understand the mechanism of action, because we don't know the single dominant gene that's responsible for this resistance. And there's a U.S. utility patent to be had there if we can prove it first. So Sammy is doing transcriptomics and going back to the basic science to use as a tool to try to get this information for us, and then ultimately engineer plants to prove the mechanism of action of the genes that she's looking for. So that said, we now have partners, Proven Winners and Bonnie Plants. Bonnie is the largest herb grower in the world, and 4% of their business is basil. They want their new inbred lines. We're now down to BC1S5. And so it hasn't been cheap to get there. Here's the lines on the right. Caesar is the control. These were inoculated with downy mildew, as were the 1965 plants on the right. So we've got the flavor and we've got everything. We've just been able to reconstruct basil with the genes from that wild relative. So the take home on all of this for today's talk is this: you really can have all three, okay? And your job is to do this and make this happen. You can have good, fast, and cheap, but you're going to have to do it by managing all your assets well. You've got to remain flexible and roll with the technology and use the technology appropriately, and keep on learning those new tools, because you never know when you're going to need them. I haven't done transcriptomics in several years, and now we're just pulling it right back out of our toolbox and using it again, and it's an improved technology. So with that said, I appreciate your time today. I do support the Collegiate Plant Initiative. That's an organization of students that help get college students exposed to jobs in plants. And if you want to learn any more about them, they're at Collegiate Plant Initiative. And then if you want to see more about the Coleus program, you can see us at the Coleus King on Instagram. So thank you very much. And I don't know if I have time for questions, but I'll turn it back over to Julian.
Okay, so hi everybody, my name is Eleni and I am a PhD student in the Horticultural Science Department. And first of all, thank you, Dr. Clark, for your very interesting presentation. We have time for 2 questions. I can guarantee that I'm not the only one that felt that is interesting because we have a lot of questions, but we're gonna go over 2. So
Okay.
the first of them is, how did you choose the initial 4 crops to screen for potential breeding?
I let the graduate student choose what she liked, because to me, I didn't really care. We were starting a new breeding program and I was just there to watch. And so I realized that if people like what they're doing, then they're going to be a lot more successful at it. So I let her have free reign, and she happened to like the Lamiaceae. And so 3 of those crops were Lamiaceae, and one that I threw in there was the bulb crop, and that was the stupidest decision, was the one that I made. So she did all of that. Absolutely.
Okay, awesome. We have another question that says, is there any breeding program or possibility of working on hydrangeas?
Yes, actually, my colleague at North Carolina State, Tom Ranney, he is at the Mountain Home location there in Asheville, which is a branch campus. He breeds some of the most fabulous hydrangeas. And also a former Gator, John Reuter at the University of Georgia, works on hydrangeas as well. So that's a big focus in the woody plant world, is hydrangea. So if anybody's interested in that and wants to learn more, I can get them connected to the people that do that.
Awesome, great. So now we have some more questions, but Dr. Clark can answer personally to them and via text, as we need to move on to our next section.
Yeah.
Thank you again, Dr. Clark. Great
No problem. Thank you. I hope you guys have a great day.
You too.
Yeah, thank you, Dr. Clark, for your presentation. I'm now going to actually introduce our first student speaker of the day. And it's no secret as to why the student was selected to give a presentation today. She's demonstrated an effectiveness as a science communicator by winning numerous competitions, including 3-minute thesis competitions organized by the National Association of Plant Breeders, flash talk competition organized by the UF Environmental Horticulture Department, oral presentations at Syngenta Plant Breeding Retreat, and more. Swati Shrestha is a PhD student in the Vegetable Breeding and Genetics Lab here at UF IFAS Tropical Research and Education Center, located in Homestead, Florida. She's currently working on the development of high-yielding virus-resistant squash cultivars using traditional and molecular breeding approaches. Here to give you a more detail on how her project is going, I'm excited to introduce Swati. Go ahead, Swati.
Thank you, Julian. I'll just share my screen first. Okay. Can you guys see my screen?
Yes, we can.
Okay, just a sec. I'll do it to a pointer view. Great. Good morning, everyone. First of all, I would like to thank Plant Science Council for organizing this amazing symposium. And I must say I'm honored to be one of the student speakers in the symposium. So as Julian mentioned, I'm Swati Shrestha, currently a PhD student with Dr. Jeffrey Meru in Tropical Research and Education Center, Homestead. And I'm excited to talk about my research in front of you all. So the topic of my talk today is identifying quantitative trait loci and developing diagnostic markers associated with zucchini yellow mosaic virus resistance in squash. And this is the project I have been working on since last 2 years. To begin with, a little background on the genus Cucurbita. The Cucurbita genus includes squash and pumpkin, and as you can see here in this picture, it's a diverse phenotype, which includes 27 different species, out of which 5 are the cultivated ones. And the squash and pumpkin are grown in most of the subtropical and temperate regions of the world. As you can see here, they have versatile use ranging from pies to soups to lattes. They can also be used as processed foods, and the recent studies have shown that they have anti-cancerous and antioxidant property, which is really cool. And squash and pumpkin can also be used as ornamental crops. They have decorative value, so it has a versatile use, as I said. Now, out of the 5 cultivated cucurbita species, 3 are the most economically important ones, which includes Cucurbita pepo, that are the field pumpkins and zucchini, Cucurbita moschata, which includes the butternut squash, crookneck, and calabaza, and Cucurbita maxima, which are the giant pumpkins. All these 3 species include both summer and winter squash, so they can be grown in both summer and winter. Now, a little bit on squash production in the US. Here in this map, the green shaded states are the ones which are the major producer of squash and pumpkin. And this darker green shaded state is Florida, where we are located, and we carry out our trials. So in the year 2019, the planted and harvested acres of squash and pumpkin was like this. And here we can see that Florida was ranked second in US after Michigan in terms of planting and harvesting of fresh market squash. Similarly, in terms of values, Florida was ranked third after California and Michigan in terms of the value of the produced squash and pumpkin. So from this, we can say that Florida is one of the major squash and pumpkin producing states in the US. Now, as we all know that crop and disease are one of the major yield limiting factor in any crop production system, and squash and pumpkin are not an exception to it. In case of squash and pumpkin, zucchini yellow mosaic virus, or ZYMV, is one of the devastating viral disease, which is an aphid transmitted virus. And the zucchini yellow mosaic virus pressure is higher in Florida, mostly because of its hot and humid weather condition, which allows for the rapid multiplication of the insect vector population. Some of the symptoms of ZYMV include severe leaf mottling, as can be seen here in this picture, and it also causes deformed fruits, as can be seen here in these pictures. Many of the farmers here in Florida has reported that cultural and chemical practices for controlling ZYMV are resource intensive and not fully effective. So a good option for managing zucchini yellow mosaic virus in squash and pumpkin would be adoption of virus-resistant cultivars. However, unfortunately, until now, only a few cultivars resistant to ZYMV are commercially available, and their resistance is not very stable. So the virus-resistant cultivars can either be produced by using traditional breeding program, where the phenotypic assays for disease resistance are tedious and resource intensive, or the virus-resistant cultivars can be produced by using molecular breeding program, where the virus-resistant cultivars can be or virus-resistant individuals can be selected by using diagnostic molecular markers, and thus greatly reduces the time required for the development of the new cultivar. Previous studies in both agronomic and horticultural crops have shown that QTL-seq based on whole genome resequencing is a powerful tool in developing resources for marker-assisted selection. So based on this background knowledge, the objective of the study that I did was to identify the QTLs associated with ZYMV resistant in squash and pumpkin using whole genome sequencing based QTL-seq method, and develop molecular markers associated with ZYMV resistance in squash. So for the study, the first step was the development of the mapping population. And for this, we used the ZYMV resistant low-yielding local landrace from Nigeria called as Nigerian local as the female parent, and ZYMV susceptible high-yielding commercial cultivar called as butterbush was used as the male parent. So these two parents were crossed to produce the F1, and then the F1s were self to produce the F2. The F2 plants were then mechanically inoculated with the natural strain of ZYMV to check their segregation to the ZYMV virus. And on the basis of the response of the F2 population to the ZYMV virus, we selected 10 most resistant individuals and called them as the resistant bulk, and selected the 10 most susceptible individuals and called them as the susceptible bulk. Now, absence of the ZYMV virus in the susceptible bulk and presence of the ZYMV virus, absence of the ZYMV virus in the resistant bulk and presence of ZYMV virus in the susceptible bulk was confirmed by using ELISA test. Now, after getting the resistant bulk, susceptible bulk, and the parents, we would move into the whole genome sequencing and the QTL analysis, and the workflow for this is as follows. First, we did the DNA extraction from the parents, the resistant and the susceptible bulk. Then the DNA were sent for sequencing. After getting the sequencing results, we align the parents to the reference Cucurbita moschata genome using Burroughs wheel aligner to develop a consensus sequence. Now, the consensus sequence was compared with the resistant and the susceptible bulk genome using the GATKit software to get the SNPs that are different between the resistant and the susceptible bulk. Now, the SNPs obtained from GATKit was transferred into the QTL-seq-R software for the QTL analysis and QTL identification. After identification of the QTLs, the markers were developed using the batch primer 3 and primer 3 plus software in those specific QTL regions. So moving towards the most interesting parts, that is the result of the study. A total of 174 F2 plants were mechanically inoculated with the natural strain of the ZYMV virus. And here in this figure, I have shown how the susceptible F2 plants look like as compared to the Nigerian local, which was the resistant parent, and how the resistant F2 individuals look like. So here we can see that the susceptible F2s were really stunted and the resistant F2 did pretty good. They looked really healthy in the screening. Out of the total 174 F2 individuals, 55% were the resistant ones and 45% showed susceptibility to the ZYMV virus. Now the results of the QTL analysis. This table here just shows how the sequencing data looked like, the quality of the sequencing data looked like after we got the sequencing results. Briefly, the mapping rate varied from 94 to 98%, which is pretty good for resequencing. And the final effective mapping depth ranged from 81 to 101, which was also really good. As I mentioned in the materials and methods, we use the GATKit software to detect the SNPs between the resistant and the susceptible bulk. And actually, a total of around 1 million SNPs were identified. So our next job was to transfer these SNPs into the QTL-seq-R software and then use these SNPs for the QTL-seq analysis. So basically, the QTL-seq-R software, it filtered out a lot of false SNPs based mainly on the quality of the SNPs, and we were left with a total of 628,000 SNPs for the QTL analysis. The results of the QTL analysis showed us that significant QTLs associated with ZYMV resistance were present in chromosome 2, 4, 8, and 20. And before I jump into this graph, a little background on this graph and whole genome resequencing based QTL analysis in general. So basically in whole genome resequencing, what we consider is that the genomic region of the susceptible and the resistant population is same across all the regions, except the region controlling the disease resistance. And in our case, the genomic region similarity or differences was measured in terms of the SNP index. Here, this graph shows the SNP index of chromosome 2, 4, 8, and 20 in susceptible bulk. This graph here shows the SNP index of the resistant bulk, and this graph here shows the delta SNP index, that is the difference in the SNP index between the resistant and the susceptible bulk. And this blue line here is the 95% confidence interval, and the red line here is the 99% confidence interval. So the region here inside the red box, these are the regions that were significantly different between the resistant and the susceptible bulk. So we can say that these are the regions significantly associated with CYMV resistance. Similarly, the region here in chromosome 4, here in chromosome 8, and here in chromosome 20 shows that these regions were significantly different between the resistant and the susceptible bulk, or in other words, these are the QTLs associated with CYMV resistance. Now, after identifying the QTLs associated with CYMV resistance, our next step was to develop diagnostic markers around those QTL regions that could easily discriminate among the resistant and the susceptible individuals. So for that, we developed 12 indel markers and 20 SNPs marker from those 4 chromosomes, and then tested these in the F2 individuals and the parents to see if there is any significant association. And interestingly, one of the indels from chromosome 20, 2 Cas from chromosome 20, and 1 Cas from chromosome 8 were able to significantly differentiate the resistant and the susceptible individuals at the molecular level. The association in the F2 population ranged from 78 to 87%. Here in this figure, I have just shown how the amplification looked like with both indel and the Cas marker. So here we can see that in case of the indel 2, which was significantly associated with the CYMV resistance, the butterbush, which was the susceptible parent, didn't show any band, and as accepted, the susceptible bulk also did not show any bands, which means that there was no amplification in the susceptible population. However, the Nigerian local, which was the resistant parent, and also the resistant bulk showed these beautiful bands. So the results of the CASP, here's the result of the CASP marker for just one of the CASP marker. Here we can see that the marker was also able to differentiate the resistant population and the susceptible population, and also the F1s, that is the heterozygous. And the other 2 CASP marker also showed similar results, but I have not included them here in this presentation. But from this, we can say that we were able to develop the indel and CASP marker that could easily differentiate the resistant and the susceptible individuals at the molecular level. Now, this final slide, that is my take home from the study and the future implications. So from this study, we can say that the QTL-seq method was efficient in identifying QTLs associated with CYMV resistance and squash and pumpkin. And also we were able to develop indel and CASP marker that can be used for rapid discrimination of the resistant and the susceptible individuals at the molecular level, and it could accelerate the future resistance breeding program. And the study can also act as an entry point for the future gene expression and the validation studies. With this, I would like to thank the funding sources of the research, my lab mates, my committee members for always guiding me. And I would like to thank you all for attention. And if there is time for questions, I'm ready for that.
Hi, Swati. Thank you so much for your great presentation. So we have one question here. How does variability in environment season affect the disease resistance? Did you test that yet?
It does affect, but in our case, all the experiments were performed in the greenhouse inside the cages. So it removed the environmental variability factor a lot, I guess. And we do inoculate them mechanically. So all the plants are supposed to get the disease if they are susceptible.
Oh, okay, that's great. And what are the next steps that you see for this project, and what do you see for the future?
So our ultimate goal, I would say, is the release of cultivar that would be resistant to CYMV. So right now I'm doing some back process, but then we are using the markers to select the resistant population. So we just hope that it will speed up the entire process and then we'll be able to release the cultivar soon. I hope so. Fingers crossed.
Okay, thank you so much for your presentation.
Thank you.
Thank you,
I'll
Swati,
stop
for your presentation.
sharing.
I'm now going to introduce our next student speaker. Our next speaker is a fourth-year PhD candidate in the Plant Molecular and Cellular Biology program here at the University of Florida. Christina Finnegan received her bachelor's degree from UF, where she worked as an undergrad research assistant developing molecular markers for breeding of tomatoes. She soon discovered that, like many of us, she had a sweet tooth and tomatoes weren't quite cutting it for her. Christina is now a member of the Sweet Corn Breeding and Genomics Lab, working with Dr. Marcio Resende as she uses quantitative genetics to study nitrogen use efficiency. She's also involved in a project that focuses on engineering symbiotic nitrogen fixation in crops. Her work brings quantitative genetics, molecular biology, and bioinformatics together to accelerate and improve plant breeding. I don't think I can wait any longer to hear her presentation, so with that, may I introduce Christina Finnegan.
Hi everyone, can everyone see my screen and hear me okay?
Looks good.
All right, thank you. So thank you, Julian, for the introduction. Like you said, my name is Christina Finnegan. I am a PhD candidate at the University of Florida in the Plant Molecular and Cellular Biology program, and I work in the Sweet Corn Breeding and Genomics Lab. Today, I'm super excited to tell you guys about a project I've been working on, characterizing the transcriptome dynamics in sweet corn lines. So for anyone who might not be familiar, sweet corn, which is the kind of corn that you would buy at a grocery store, at least if you live in the United States and make corn on the cob to eat fresh, whereas field corn is the kind of corn that you hear about us making ethanol with, livestock feed, etc. There are a number of differences between these types of corn, but first and foremost, sweet corn has a buildup of sugar in the kernels, because of mutations in the starch biosynthetic pathway. This gives it its sweet taste, which is why we like it. So the reason this corn is sweet is mutations in the starch biosynthetic pathway. And there are a number of mutations in this pathway, but only a couple of them are really widely used as far as sweet corn cultivar development. So I'm gonna give a quick introduction to those just to make sure everyone is on the same page. First, there is Shrunken-2, and this is named for its kernel phenotype. You can see here. The dried down kernels are way smaller. They're wrinkly because they aren't filled with starch like the wild type or field corn kernels. What this mutation is a mutation in the endosperm ADP glucose pyrophosphorylase, or AGPase. And this enzyme catalyzes the first step of the starch biosynthetic process, taking glucose 1-phosphate to ADP glucose, which is the actual substrate for starch biosynthesis. Here I'm showing it on a diagram of the starch biosynthetic pathway. You can see it's right at the top. And so as you can imagine, this mutation has a really dramatic impact on the starch and sugar levels, bringing starch down to about 25% of the wild type starch levels, and the sugar goes up to counter that. There's also sugary 1, which is further down in the starch biosynthetic pathway. It's a mutation in isoamylase 1, a starch debranching enzyme. And so the phenotype is not as dramatic, but you still get that decrease in starch and increase in sugars. So just for everyone's information, in general, although it is subjective, the more sugar in a sweet corn variety, the more liked it is by consumers, and then therefore, the more the breeder wants to continue producing that line. So in addition to these two main mutants, there are a number of modifiers that breeders use in conjunction with these to further bump up those sugar levels and keep make the taste good for consumers. One of these is called Sugar Enhancer 1. So this is a mutant that we don't actually know mechanistically what it's doing, but we do know that it bumps up the sugar levels in sugary 1 backgrounds. And it was recently mapped to a gene in 2019 by the Kepler group, but we still don't have an idea of what it might be doing and how it might be affecting metabolism. So what we set out to do in this project is understand regulators of the starch biosynthetic pathway. You can think of those two main mutants that I mentioned as ways to rock the boat on starch metabolism. And you can kind of think about them the same way as you can subjecting a plant to a heat stress or whatever you're interested in. So we want to see what else changes in the genome and what's driving those changes. For both basic reasons, we want to understand the regulation of this pathway. We want to learn more about the modifiers that we know exist, but we don't know how they're working. But we also want to understand this from an applied sense point of view. If we can identify potential regulators that can bump up the sugar in these already existing mutant lines, those become targets for breeders who want to increase the quality and sugar levels in their lines. So it's kind of a dual approach here. So in addition to the mutants I talked about, there's also something called background effects that breeders have to keep in mind, which means that the line or background a mutant is in can further affect the phenotype. And so to make sure that we capture that in this experiment, we use near isogenic lines contributed by Bill Tracy at the University of Wisconsin-Madison. We have 6 different genetic backgrounds, and for each of those, we have a wild type line where there are no mutations in the starch pathway, sugary 1 line, and a shrunken 2 line. And so we planted these in Belgrade, Florida, and then we grew them over the course of the season. And we harvested at different developmental time points to get an idea of how these mutations affect the transcriptome over time. We harvested at 14 days after pollination, which is right at the beginning of starch biosynthesis. It's called blister stage. 21 days after pollination, which in a wild type kernel would be when starch biosynthesis is really active. But this is also when sweet corn is harvested for consumption. 28 days after pollination, these kernels have pretty much completed grain filling and they're starting to dry down. And then in addition to those developmental time points, we also saved some of the ears that we harvested at 21 days after pollination, and we kept them in the refrigerator for 7 days to mimic the storage and shipping process that growers have to do to actually get their ears to the consumer. And this is for us to gain a little bit of insight into the genetic control of varying shelf life and post-harvest quality. So we had multiple reps of each of these treatment combinations, and then we pooled them. Some of these kernels we used for the actual RNA extraction, and I dissected these kernels and specifically extracted RNA from the endosperm. And that's because this is the main starch and synthesis and storage enzyme, or storage organ. But we wanted to also avoid the confounding effects of, say, the embryo developing over time. So we extracted from the endosperm and performed differential expression and co-expression network analysis to look at genes behavior. We also use some of these kernels instead for wet lab quantification of the actual starch and sugar levels. And we can then perform analysis linking the gene behavior to the phenotypes that we see in the kernel. So now I'm going to jump right into the results. So the first thing that we wanted to look at was a principal component analysis of the transcriptome. And what this does is it basically looks at all of the data for all of the genes, for all of the samples, and reduces it to this single graph. And this allows us to find major trends that are driving the transcriptome. So right here, you can see that time point is the main driver of these clusters, which means that time point is the most important thing to what a transcriptome looks like on the whole. You can also see here I've just colored by the actual mutant. You can see that there's kind of a grouping of shrunken 2 with this peachy color, but the sugary 1 and wild type transcriptomes seem really similar. And this kind of gives us a hint that the shrunken 2 transcriptomes are a lot more different from the wild type, but the sugary 1, which is that mutation further down in the pathway, doesn't change the transcriptome as much. And so this kind of informed our future analysis. We split the data by time point rather than merging everything together to avoid losing some of those effects that might only happen at one time. So the first thing we wanted to look at was general changes between the shrunken 2 endosperm and the wild type. So overall, we see a downregulation of starch synthases and starch degradation enzymes. And this makes sense because neither of those enzyme groups have their typical substrates, which is ADP glucose for the starch synthases, and then the starch itself for the starch degradation enzymes. We also see a downregulation of certain sucrose transporters, specifically SUT1. and trehalose 6-phosphate synthetases. And so these enzymes create trehalose 6-phosphate synthase, or trehalose 6-phosphate, which is an important signaling molecule for a variety of processes. It's been implicated in drought stress, sugar signaling, carbohydrate and nitrogen balancing pathways. And you'll notice that this is matched by an increase in trehalose 6-phosphate phosphatases, which degrade this molecule. And so this implicates that maybe in the shrunken 2 endosperm, the pool of trehalose 6-phosphate is being actively reduced. We see an upregulation of sugar exporters, specifically of the sweet family, and an upregulation of the embryo version of shrunken 2. We see an upregulation of heat stress and wound-induced proteins. And we think this is just a byproduct of the buildup glucose in the kernels because vacuolar glucose is associated with heat and wound stress. So like I mentioned, we are not only interested in the changes between these transcriptomes, but we want to find those key drivers of the change or the regulators. And so what we did was perform weighted gene co-expression network analysis, or WGCNA. And what this does is it looks at the gene expression of all of the genes across all of the treatments. So that would be our shrunken 2 versus wild type, the various genetic backgrounds, and across time points. And it looks for genes whose expression is correlated with one another. And that indicates to us that they are co-regulated. And so they are put into modules, and every gene, every line that you see on this tree represents a gene. And then down here, these colors represent what group that gene has been put into. We can then perform analyses where we match these groups of genes to the phenotypes. So what you can see here is that, for example, this red module is really positively associated with sucrose levels. And so we might look into that to find out what is really driving that increase in sucrose. And these phenotypes are controlled for shrunken 2 or the sugary 1. So this means that there's more sucrose even if you just compared like the shrunken 2 samples. So like I mentioned at the beginning, one of the things that we're really interested in is finding more clues as to what these modifiers that we know are important, but we don't know what they're doing, finding clues about their actual role. So we actually found that sugary enhancer 1 is a hub gene or potential regulator of one of these modules. And this is really exciting because it was just recently mapped in 2019. We know sugary enhancer 1 has a role in starch metabolism, but we don't really have any idea as to what it might be. And so we actually found that it appears to be a regulator of a number of important developmental transcription factors, including opaque 7, which is involved in grain filling, scarecrow-like 1, and a number of other GRAS transcription factor family members. We also see a number of polysaccharide degradation genes in this module. And this makes a lot of sense because in the sugary enhancer 1 mutant, we see an increase in maltose, which is a result of starch breakdown. And so while we still don't know exactly what sugary enhancer 1 is doing as a wild type gene in its functional form, this gives us a lot more insight into directions we can pursue for future research, but it still coincides pretty nicely with the things that we do know about it. So far, everything I've talked about has been a result of merging all of the different backgrounds together. But like I mentioned at the beginning, not all of these backgrounds respond the same to these mutations. So what I'm showing here is the total sugar content at 21 days after pollination, which is when these sweet corn lines would be harvested theoretically. So you can see that some of these genotypes, like this Iowa 453 and 5125, show a really dramatic increase in sugar levels from the wild type to the shrunken 2. Whereas others, like C40 and this IL-101T, the sugar levels still increase in the shrunken 2 lines, but not by nearly as much. And so we wanted to look at genes that are associated with this pattern of responsiveness. Some genes not only varied in how much they were differentially expressed, they actually varied in direction. So these two histones, H2B5 and H2A6, are actually downregulated in the non-responsive genotypes and upregulated in the more responsive genotypes, IO453 and 5125. And so this indicates that epigenetic regulation might be underlying these background effects that we're seeing and might be limiting breeders in how sweet they can get sweet corn in certain backgrounds. We also mentioned, I mentioned at the beginning, we saved some of these ears in the fridge to look at post-harvest effect. So we measured the sugars at 21 days after pollination, those fresh ears, and then after storing them in the refrigerator for a week. This is really important because when you buy sweet corn, it has been stored and shipped. And so the final product that the consumers are getting isn't necessarily the sweetest corn that they could be getting. So we looked at the differences in sugars post-harvest. Iowa 453, for example, loses a lot of sugars over the course of that week, where C68 and 5125 lose very little, and then in this case, significantly, there's no difference. So we looked at genes that are also associated with this pattern. We found a transcription factor and a transporter that are upregulated in these genotypes that don't lose as much sugar. So these become potential breeding targets for breeders who want to focus on shelf life and post-harvest quality in their lines. So just to wrap up, we set out to learn more about the regulators of the starch biosynthetic pathway and also the modifiers that we know exist. And it turns out those are kind of the same thing. Sugary enhancer 1 is a regulator of a network containing really important transcription factors. And so this is going to guide future work trying to really nail down what this gene is doing and how it's affecting starch biosynthesis. We also identified potential modifiers for breeders. We have these histones that are affecting the sugar levels in shrunken 2 lines, as well as transcription factors and transporters that are affecting the post-harvest quality. So in the future, we plan on following up these modifiers that I've mentioned and just making sure that these patterns really do hold up, and then eventually incorporate those into our breeding program to improve the sweet corn lines at the University of Florida. And so with that, I would like to thank everyone in the Resende lab as well as the Tracy lab for contributing the near isogenic lines that made this study even possible. So with that, I will take any questions.
Thank you, Christina, for the question, for such a wonderful presentations. And with this, we are very happy to take any questions. Please feel free to type them in the Q&A part. And meanwhile, I do have some more questions that I would like to read out. So we have a question from Julian, and he's asking, what does your future work include? And do you see a cultivar being released anytime soon?
So as far as future work, we have a lot more of these co-expression modules than just the one that Sugary Enhancer is the regulator of. And so I'm really interested in diving further into those and the other regulators and seeing what they look like they might be doing and the role they might be playing in starch biosynthesis. As far as actually targeting these in the breeding program, this is pretty preliminary work as far as going for it in a breeding program, because that's a lot of investment. So we would have to do a lot of validation before these made their way into a cultivar.
There's another question from Scott, and he's asking, how would you use this data in an applied sweet corn breeding program to select or breed for enhanced flavor and improved sweet corn varieties?
So I think that if this led to understanding the role that Sugary Enhancer 1 is playing, we could try to find other genes like it to introduce variety into the modifiers we can use. Variety is the heart of a breeding program. And so if we know what it's doing, we can search for other things like it. And then, for example, if we find that the epigenetic regulation is really limiting the lines that we can put shrunken 2 in and get sweet corn, if we can then target that by breeding, somehow breeding that out or breaking those associations, we then have a whole another set of varieties that we can incorporate into breeding programs, which may have other good qualities that we don't know about because we can't get that sweet flavor yet.
Thank you. And we have another question by Fade, and he's asking, are you interested or looking into in terms of regulatory sequences that may influence the starch biosynthesis based on background diversity?
So are you asking, I think they're asking about looking into like cis promoters that are popping up. Is that the question?
Possibly, yeah. It does mention about regulatory sequences there.
So I think that would be really interesting, especially if we could find that all of the genes in one of the modules had like a common cis regulatory element. That would really help us narrow down how these regulators are working. But as far as applied applications of that, I don't know off the top of my head.
Thank you. And we have another more question. That's what was the most challenging part of your research? And this is by Bonnie.
The most challenging part, I think, is sifting through the amount of data that you get from an experiment like this. So we had 6 genetic backgrounds. For each of those, we had 3 lines, wild type, sugary 1, and shrunken 2. And then we had 4 different time points. And so you can imagine that the amount of information you get is really intense. And then you have to sift through it to find the parts that both make sense with what we do know, but also provide new information. And so I'm still working on that. And it's definitely the hardest part.
Thank you. And there's one more question by Modesta, and she's asking, does any of this sugar trait affect yield and how?
Can you say that again?
Does any of this sugar trait affect the yield and how, if it does?
So it does affect yield. And then in sweet corn, we not only are considering yield, but marketable yield. And so it has to be something that tastes good, otherwise it's kind of useless. It also affects yield really directly. The less starch there is in a kernel, the harder it is to get those fields to germinate. And so sometimes breeders run into this issue of there's a lot of sugar, people really like it, but a really large percentage of the field they plant doesn't germinate. That's a loss for the growers, so you can't tolerate that. So I think that.
Thank you. And we have one more question. So the question is by Gabriel, and he's asking, do the modifiers also modify the overall agronomic traits, in the different backgrounds like disease resistance, vigor, and yield?
We don't know that. We don't have information from this field trial on things like that, but it would be interesting to follow up maybe in like a lit search to see if they're controlling things like that.
Cool. And most likely we might have one more question by Joshua, and he's asking, sweetness is a good goal. I like sweet, but how about making nutrition more available for our bodies? So we might not need to do nixtamalization to make the nutrients available.
I mean, that's an excellent point. That's an excellent goal. As far as consumer liking and sales, sweet is still the most important factor for sweet corn. But you're right. I don't know exactly how to go about that in sweet corn because most of the calories in sweet corn are from the sugar. So.
Thank you. So with this, we are done with the Q&A session, and Julian will be taking over. Thank you.
Thank you, Christina, for your talk.
Thank
I'm
you.
going to introduce our last student speaker of the day, and I could start this introduction by listing off her research achievements, but I think it would be equally as important to mention all of her outreach endeavors. Samantha Burrell has a knack for science communication and has co-founded a K-12 plant science outreach program called Exploring Plants, Growing Under the Rainbow. She's won the UF 3-minute thesis competition and co-authored a college gardening textbook submitted to the UF Publishing Office titled Plants, Gardening, and You. As a PhD student in Dr. David Clark's lab, Samantha's research project focuses on discovering how the new basil cultivars produced by UF's basil breeding program resist basil downy mildew. She hopes to pursue a career in plant biochemistry and molecular genetics focused on plants for medicinal and human health, and continues to participate in science communication. With that, I'm happy to introduce Samantha Burrell.
Thank you for that introduction, Julian. I'm going to go ahead and share my screen. Is the speaker show, or is it the actual?
We see the notes version.
Okay. There. Is it corrected?
That's perfect.
Wonderful. Okay, thank you again for such a wonderful introduction. I'm very happy to be here at the 5th Annual Plant Science Symposium at UF. I'm going to be talking about my research working with Dr. Clark and the UF Basil Breeding Program, and focusing on how their amazing amazel cultivars are able to resist basil downy mildew. Sweet basil is the most popular herb in the world, and US markets alone gross over $300 million annually. Major growers in the US produce about 100,000 pounds of basil per week. That's a lot of sweet basil. Let's see, to put it pictorially, that's if every single one of these basil plants was 1,000 pounds. This is how much that major growers are producing each week. In 2007, basil downy mildew came into the US through South Florida and quickly spread throughout the country, causing major crop loss. And it actually dropped the weekly production from 100,000 pounds to only 10,000 pounds a week. That's a 10-fold drop in production, and it's huge. It's decimated the basil industry. BDM looks like a yellowing on the leaf surface, and as the disease progresses, it starts to just completely destroy the leaf, which is the marketable part of the plant. And finally, sporangia will emerge on the bottom side of the leaf. This disease is caused by Peronospora balbari, which is an obligate oomycete. It's a biotrophic pathogen, and under a microscope, it looks like this. These little circles are the sporangia, and this germ-like structure is a secondary hyphae. So this is what would emerge from the leaf tissue to produce all of the sporangia on the bottom. So the UF Basil Breeding Program was born to combat this disease. And, you know, it came into the game when no existing resistance existed, no existing resistance in sweet basil. So they had to look to other species of basil for outcrossing. Basil species are promiscuous, if you will. And they were able to find a good source of resistance in Ocimum americanum, which is, it's like a lemon basil. It's a wild species of basil from Africa. This one key cross was made between regular sweet basil, Ocimum basilicum, and Ocimum americanum in order to produce an interspecific F1 hybrid that had complete resistance to downy mildew. Here are the results of a BDM screen, and you can see both of the parents below. The susceptible is completely decimated. The resistant looks fabulous, and so does the interspecific F1. So the F1 species went on to years of back crossing with sweet basil and inbreeding to get more of those desirable sweet basil traits. And the UF Basil Breeding Program was able to produce the first Italian sweet basil resistant to downy mildew, otherwise known as amazel basil. Here's a picture of the industry standard sweet basil called Italian large leaf next to amazel, and clearly I think you can see that it was a success. Downy mildew is truly not welcome on this plant. So it's wonderful now that there are basil cultivars that are resistant to this plant disease. However, the mechanism of resistance is still unknown, and that's the question that I'm trying to answer. So I can take advantage of the basil breeding lines, and I chose a family called 19-22 that is bred for homogeneity of traits, but still segregating for downy mildew resistance. And this is the result of another BDM screen where you can see that the resistant background looks great starting out, so does the susceptible. However, at 10 days post-inoculation, you start to see a difference between these two sibling basil plants. The resistant one still looks wonderful. You cut it off and you look at the underside and there's nothing there, but the susceptible is definitely showing the signs. You see the yellowing of the leaf surface, you cut it off and you see the sporangia is definitely emerging. So I wanted to take my observations to a more microscopic scale to get a closer look, pun fully intended. And I was able to do so using epifluorescent microscopy. And I saw that at 24 hours post-inoculation in the resistant plant, you can see the sporangia of the pathogen has grown a germ tube, grown an aprosorium-like structure, and penetrated through the host tissue, showing a production of callose by the plant. This production of callose extends further than just the aprosorium, and even by 48 hours post-inoculation, you see this callose production. It's very large, indicating a host response to the pathogen penetration. In the susceptible background, an entirely different story is going on. The sporangia is still forming a germ tube in aprosorium, and you see that it penetrates with a little callose production. And then you start to see that the pathogen colonizes the inside of the host cell pretty rapidly, and I would even say aggressively. Each of these white arrows indicates a hostorial knob that the pathogen has formed to start sucking out the nutrients from the plant and survive. And even at 48 hours post-inoculation, this colonization has really taken off throughout the entire plant. So now that I knew the differences occurred at the 24-hour post-inoculation point, I wanted to go ahead and compare the plant pathogen interactions at a level of gene expression. So I did a de novo transcriptome assembly and a differential gene expression analysis, and I compared both of these backgrounds at that 24-hour post-inoculation time point versus a control, so just before, like, no inoculation. And then I also compared them to each other at the 24-hour post-inoculation time point to see what kind of cellular machinery was being turned on and turned off, and what was the big difference. So first, I chose the resistant background inoculated, so RI, versus the resistant background control. And I found 274 total differentially expressed genes. That's a pretty low number of differentially expressed genes, but I guess, you know, these backgrounds are so similar and only a few genes may have been turned on for this response. And next I went ahead and did a Go enrichment analysis of those genes. And I saw that in the upregulated differentially expressed genes, that means that these are ones that are more highly expressed in the resistant inoculated than the resistant control. There is a huge enrichment for the response to biotic stimulus. This is a tree diagram in which the size of each box corresponds to the magnitude of the enrichment that the term had. So response to biotic stimulus, defense response, the ABA signaling pathways being turned on, DNA transcription in response to stress, response to wounding, response to molecule of fungal organism. It's very apparent that the resistant plant is trying to fight off the pathogen. And that corresponds pretty well with the microscopy showing the host response at this time point. Also, like you can see, response to biotic stimulus, that this box is so large, that is a much higher response than something like the pyruvate transmembrane transport, which was also enriched, but just not to the same extent. We also see some chitin catabolism going on, and I think this probably has to do with the response that we're seeing as well, where the cell wall of the plant and the pathogen are kind of battling it out. So analyzing the downregulated genes, so these are the ones that the inoculated plant is not expressing as highly as the control plant is. We see like ATP metabolism was probably shut off so that the defense response could be upregulated. And you just see like regular things that the plant would be doing that it could maybe sacrifice a little bit of that gene expression in order to go ahead and kill that pathogen off. Next, I compared the inoculated susceptible background with the control in the susceptible background, and I found, again, a small number, but 231 total differentially expressed genes. 164 were up and 67 were down, in case you were wondering. And again, going into the Go enrichment analysis and this tree diagram, we see that the TCA cycle is very upregulated. Somatic embryogenesis, a little bit of sesquiterpene biosynthesis, secretion, which was really interesting. And keep in mind that this inoculated sample also includes whatever the pathogen transcriptome could have been sequenced. So some of these might even be like this beta-glucan biosynthetic process. That makes sense for it to be one of the pathogen genes because those germ tubes and those hyphae structures are made of beta-glucans in the oomycetes family. So we're starting to see maybe the pathogen is starting to colonize through. It totally corresponds with what we were seeing with the microscopy. And it doesn't look like the plant is even reacting, like it might be completely blinded to what's going on according to its gene expression. In the downregulated ones, we see other things like chitin catabolism. So maybe this, in fact, does have to do with defense response. Not sure because it's in both. And we see other things like wax biosynthesis. Response to wounding is downregulated. So maybe even this pathogen is essentially blinding the plant with some sort of effector signaling process. So finally, I just wanted to look at the inoculated resistant versus the inoculated susceptible to try to see what genes are really responsible for this resistance. And I was able to produce a list of really good candidate resistance genes that were upregulated in the resistant versus the susceptible at this time point. I won't really go through them all, but I'm thinking maybe this chitin elicitor receptor kinase 1, that looks like a really good one to me. So that's going to be high on my list for the next steps, which will be transforming mutants to investigate those candidate genes and also characterizing the metabolic changes that occur upon inoculation. The metabolic changes are something that showed up a little bit in the tree diagram. So I wanted to see if maybe the volatiles are affected, terpenes, and possibly the flavonoids and phenolic acids. I'd like to go ahead and thank everyone that's been a part of this research, at least on some small level, and the Environmental Horticulture Department, which I am based out of in the University of Florida. And with that, that's the end of my talk. So I guess I can take questions now. Thank you all for listening.
Thank you, Samantha. So yeah, please shoot out any questions in the Q&A session. We haven't received any so far here right now, but I do have a couple that were texted. And so one question is, was there any drawback from the initial interspecific cross and any sort of linkage drag or something like that?
Oh, that's a good question. So the major drawback of the initial cross was that many of the Ocimum americanum traits had made their way into the hybrid. So the americanum plant looks a lot different from sweet basil and also smells a lot different. So those are two, like if a consumer doesn't recognize it as sweet basil, and if it doesn't even smell like sweet basil, it won't be very marketable. So that's kind of why it took several years of the back crossing and the inbreeding to make a better plant. In response to linkage drag, this gene is like a single dominant resistance gene. At least that's how it was inherited through our breeding program. So after, that initial cross, it was pretty easy to continue to get the resistance to show up in at least, half of the plants.
Thank you. We have another question, and it asks, are there any growing practices that could control mildew infestation? Like for home gardeners, will purchasing the transplants or growing from seeds make any kind of difference?
Yeah, that's a really good question, and also part of the reason why major growers' crops were decimated. There is like no treatment for basal downy mildew that is effective. Once it gets into your garden, it will take over, granted that the conditions are right, which is high humidity, especially at night. Like if there's a dew, you are going to get the sporangia coming out of your plant. The best thing that you can do to ward off downy mildew is to grow your plants in a temperature controlled environment where you are not you're not letting them get that dew overnight. But other than that, if your plant has downy mildew and you try to take a cutting and like maybe part of the plant doesn't show symptoms, it will still have symptoms later on. It's an impossible disease. It's so hard to control.
Thank you. There's another question, and it asks, how do you expect the eventual release of a trans-basil plant to be received by the general public?
Ooh, I don't know if it'll ever be received. I think it might be something that stays and dies within academia. General public does not like GMOs usually. So I don't see it as something that the UF basil breeding program will ever release. They've already got their downy mildew resistant cultivars, so why bother releasing a GMO? This is more for the basic science side.
Oh, thank you. There's another one. Have you tested the Amazon basil against other fungal pathogens?
Good question. No, we have not. I think the other bad pathogen for basil, which is not nearly as bad as downy mildew, I think is Fusarium wilt. I think is another bad one, but we have not tested it. I'm not sure how it holds up against it, and it's never been a problem for us in the greenhouse or the fields. So I'm not sure if that means that it's good against Fusarium wilt or if it's just like not come to us.
Thank you. Another one, you mentioned further research on nutritious volatiles. How will this current cross or this research inform that?
I'm sorry, can you repeat that one more time? I got a little spotty internet.
So you mentioned that further research on nutritional volatiles. So how will this current cross or this research inform about that?
So I guess you're asking more like what's the scope of the research and what am I trying to get at by looking into the volatiles? Mm-hmm. So what I would do is compare the both of the backgrounds when they're inoculated versus when they're not inoculated to see if any of the volatiles maybe are being more highly emitted during inoculation, or maybe they're not being as highly emitted, or maybe they're just not affected at all, and just try to see if there's another layer there that the pathogen may be affecting gene expression wise that then shows up biochemically in the plant.
Cool. Thank you. Yeah, I think that's all the questions that we have. So we are having a lunch break now, and we'll be back in an hour exactly. So thank you, Samantha.
So actually, sorry to interrupt there. The lunch break is from right now until 1:05 p.m. So we'll see you back at 1 o'clock Eastern time. And it's going to be on this exact same Zoom link. So thank you, Samantha, for your presentation. Thank you all who presented this morning. And we hope to see you all back for the afternoon session. Thank you.
Thank you.
Hey, Sammy. Congrats. It was a really, really great presentation. It's Gabriel here, by the way. Do you mind, would it be okay if I take a screenshot of you?
Yes, I was actually going to try to do it myself, but I couldn't figure out how to do it.
Okay, yeah, just a second, because I also need to figure out how to do that since they're on one screen. I'll try.